Hello. So Karen actually graduated from Rochester High School, but she attended the University of Mississippi biomedical engineering majors. So, and I asked her, why do you go to University of Mississippi? Do you know Dr. Mo? Dr. Mo is a faculty, retired from here and went to Mississippi as a faculty. And turn out she has a full-ride scholarship, 3 scholarships from Mississippi. So the scholarship is very generous, too much to decline. So I'm very happy that she is here as an intern in the summer. She works on this called nano-coding RNA, which is not transcribed to a protein. This non-coding RNA called RMST. At first was discovered by one of my grad students, and this RMST regulates tumor cell growth and apoptosis through called mTOR pathway. Nancy just mentioned about mTOR pathways. So, and what she wanted to study is whether this prostate cancers, when they treated by called castration therapy, enzalutamide, whether this RMST expression changes. So without further ado, yeah, Karen.
Right. Thank you, Dr. Nave, for the introduction. So my summary this past 8 weeks was about the expression of long non-coding RNA, RMST, in prostate tumor cells in response to medical castration therapy. So to define a few of those terms for y'all real quick, so LNC RNA, RMST, that stands for long non-coding RNA, which is RNA that does not translate into a protein and it's longer than 200 nucleotides. RMST is an abbreviation for rhabdomyosarcoma 2 associated transcript, it's a very long word, and it is measuring the expression of RMST in cells before and after treatment in this experiment. So I used 2 different RMST primer sets in my QPCRs. I used RMST5 mixture and an RMST7-8 mixture. You can see the gene sequences above or on the slide. Next, I would like to talk about my prostate tumor cells. So prostate cancer is cancer that forms in the male prostate gland. It is said that around 1 in 5 will actually pass away from this cancer, but 1 in 40 will be diagnosed. So it is a very late developing cancer, and it's very dangerous when we discover it this late term. So research is very necessary for this kind of cancer. I worked with LNCAP and 22RB1 cells. So you'll see the LNCAP cells over here on your far left and the 22RB1 on your right. LNCAP is short for lymph node carcinoma of the prostate, and it's derived from metastasis of human prostate lymph node lesion. 22RB1 is a carcinoma cell line from the xenograft or an implant of a tissue or organ that is different from the recipient, and it is of CWR22R cells. So you can see in the cell morphology that the LNCAP cells on the left are more rounded and they branch off in different directions, have elongated branches, and then our 22RB1 cells are going to be more pentaradial and irregular with their branching. So medical castration therapy is different than surgical castration therapy. So as Lindsay mentioned earlier, this is a hormone therapy usage. So it's a treatment for tumors that feed on sex hormones. So we are going to deprive those cell lines of the androgen. So as you all know, males produce a lot of testosterone, and these LNCAP prostate cancer cells feed on that testosterone to proliferate and to create more cancer cells. So LNCAP is an androgen receptor, and it feeds on those sex hormones. And so using these, the following drugs, we're trying to complete this medical castration therapy by depriving those of testosterone. So enzalutamide, for enzalutamide, lovastatin, and DMSO, we use a 20 micromolar concentration. So enzalutamide is most commonly used, and it is an androgen receptor antagonist, so this is the primary delivery of castration therapy. So LNCAP cells are very responsive to this kind of therapy, and 22RV1 cells are very resistant. So this has to be taken into consideration when we are seeding the cells and treating the cells, because that concentration of cells seeded and the concentration of treatment added is dependent on the number of cells. Lovastatin was also 20 micromolar concentration. It is a cholesterol-lowering medication, and it has proven decreased cell viability in LNCAP cell line. So that's some anti-tumor activity. DMSO is a dimethyl sulfoxide, and it was our control solvent. So when you're making these two kinds of drugs, you have to dissolve the powder in DMSO because it is the solvent. So when you're doing the control, you have to account for the effect of DMSO on the other cells that were treated. So I treated these cells for 6 hours and 24 hours. I ended up doing a treatment with enzalutamide, a treatment with lovastatin, and also a combined treatment. So after they are treated for 6 and 24 hours, I completed an RNA extraction procedure. So after that RNA extraction procedure, I measured the concentration of RNA for each cell harvested. So you can see all my concentrations above in nanograms per microliter. So the target concentration is around 160 nanograms per microliter, so that the concentration is viable enough to do cDNA synthesis in my next step. So cDNA is complementary DNA, which is a more stable alternative to RNA. So cDNA is a copy of messenger RNA. And we use the RNA to synthesize the cDNA. So we use the cDNA and we put this in our qPCR along with the gene primer. And my gene primers, as I mentioned before, were RMST5 and RMST78. So qPCR stands for quantitative polymerase chain reaction, and we measure CT values in a qPCR. So the CT value is the number of cycles it takes for a sample to cross the unique threshold for each qPCR. So each qPCR will have a different threshold based on the values in the qPCR. And so as you can see in this picture, the CT value of 19, that's a smaller CT value because it means the gene is highly expressed and it took less cycles to cross that threshold, whereas a larger CT value will have taken more cycles to have crossed that fluorescence threshold. The endogenous control that I used in my experiment was beta-actin. We called this the housekeeping gene. So I used this later to calculate my delta CT and to make my graphs. And it usually has a smaller CT. So when my RMST primers have a larger CT, you subtract that actin CT value. So here are some of my first qPCR results. These are LNCAP cells treated with enzalutamide, and as I said earlier, LNCAP are very sensitive to this enzalutamide treatment, and it's very apparent, as you can see here, because there's multiple statistically correct and proven numbers here of a steady increase of RMST expression after these cells have been treated for 6 hours, and then a consequent decrease for 24 hours. I also included these images that I took of my cells before and after treatment. And as you can see, before treatment, they are still in their normal cell morphology. They have rounded edges and elongated branches, and then after, they are, you can see cell death in this as they're starting to curl and lose their cell morphology, and they produce more fluorescence. These are my LNCAP cells that we treated with lovastatin. So these are also statistically correct, and these graphs show that RMST expression has increased after 6 hours, and it's either a decrease or an increase at 24 hours, but the RMST primers are different, so it wasn't consistent. But these are also my before and after treatment pictures, and as you can see, it's the same as the last slide, where in the before treatment they contain their cell morphology, and after you can see a lot of cell death and fluorescence. So this is my combination treatment that I told you all about at the beginning. So I treated them with enzalutamide and lovastatin, and you can see a significant difference in this cell death. So there is a steady increase in RMST expression in these cells after treatment. So that the combined treatment was very effective. On the other hand, we have 22RB1 cells, which I mentioned earlier, are very resistant to this kind of treatment. So as you can see, it looks like it might be steadily increasing, but I want to draw your attention to these numbers on the side that are very close to one another. So this is actually a very stagnant and unchanging value. So they hardly even increase because they are so resistant to this treatment. These are 22RB1 cells treated with lovastatin, which are similar in results to enzalutamide, a very stagnant and resistant to the cell treatment. So I ran a gel electrophoresis and a colony assay to prove these values that I got. So the gel electrophoresis is not shown above, but the gel electrophoresis basically shows that what I had and the results that I got confirmed that RMST5 was a single-banded and long non-coding RNA in my results. It was the same as the RMST5 at the beginning. And then colony formation assay is evaluates cell proliferation rates of a single cell. So basically, in this picture to the right, you can see you want to seed a very few number of cells, and then you give them around 2 weeks of growth period, and you see how they proliferated and how, and you evaluate their ability to form a cell colony. So LNCAP cells have a very slow proliferation rate, and they're very sensitive to treatment, and these cells were seeded with treatment as well. So my LNCAP cells are not included here in my results because what was expected was for them to have very small colonial growth, but however, that 2 weeks was not long enough for them to grow within the treatment, so I don't have results for those. But my 22RB1 cells have a very fast proliferation rate, and they're very resistant to treatment. So I have this 22RB1 without enzalutamide, but with a steady increase of lovastatin concentration in the cells, and you can see that their colonial growth, the number of colonies counted in each plate is a steady decrease, which is what is expected, and I'm very happy with these results. So the significance of this is, of course, that first that 22RB1 is castration resistant and LNCAP cells are castration sensitive. So RMST is a cancer stem cell regulator, and so it can be stimulated by enzalutamide specifically at the 6-hour time point because, as we saw in the other graphs, at 24 hours, the RMST expression rapidly decreased. So RMST could possibly determine tumor cell response, which is a huge contributor. So in further research, if I have the chance one day or in the future, I would like to research as if this is a byproduct or collateral of the RMST addition, or would it be used to modify cell response? So RMST could determine whether or not tumor cells respond to that treatment. So that's the end of my presentation. I would like to take a second to thank all of you donors and everybody here for coming and listening to us. I want to thank SIU and FCI, and I would like to thank my mentor, Dr. Nee, for all your help, and your grad student, Xiangwei Feng, who helped me so much and was so patient and kind with me in helping me learn all of the research this summer. Thank you.
Questions for Kaylin? I want to turn the lights up. Push the right button.
Yes.
Were the cells retrieved through prostate biopsy or how?
I was not given that information, but I know that they are from patients. So we freeze them and then I revive those cells and I use them in my experiments. Yes.
So testosterone normally declines as men age,
yes,
the levels. But now Frank Thomas is on TV every night telling us that we can take testosterone enhancers. So do you think that there's going to be more incidence potentially of prostate cancer because there are now testosterone supplements that Frank Thomas is telling everybody to take?
I definitely think that could result in more prostate cancer because I know that when men are diagnosed with heavy stages of prostate cancer at younger ages, when they do have more testosterone, it's very fatal. So if older men are taking more testosterone, they have a more likely chance of fatality with prostate cancer. So.
Yeah, quick one, Dr. B.
Do you know if your RMST interacts with the androgen receptor to stabilize it in your castration insensitive?
I'm not positive. I'm sorry.
No, it's all right.
Dr. Nee, you have a quick answer?
We don't know yet.
Here's your next aim. Look at that.
So sequence-wise, it doesn't appear to?
Yeah, it seems, yeah, we have to see the evidence. Yeah.
All right, thank you.
Thank you.